And now we're going to dig deep into plant health issues with Sarah Placella.
Having me today. Can everybody hear me all right? Okay. So today I'm going to talk to you guys about pathogen detection technologies, advantages and disadvantages. And I mean for this to be an overview of some of the techniques that you already use and then some new techniques that we are offering. So this is just some of our team, but Root Applied Sciences started in May 2018. So we're pretty young, but we're a company started by scientists that essentially we felt like there was a lot of knowledge that was common in academia and tools and techniques that you guys don't have access to. So we started this company to make them more accessible and to also help us make our work something that is useful for you guys. You know, when you're an academic, you end up always framing your research for other academics. And then it kind of gets lost to the public. So that's why we started Root Applied Sciences. I did my PhD at UC Berkeley in microbial ecology and biogeochemistry. Rodrigo is a professor of ecology and emerging infectious diseases at UC Berkeley. He's been working on grapevines for over 20 years. And Michael is our hands. He is a research scientist who specialized in plant pathogen diagnostics. A lot of you guys are probably concerned from time to time when you look at your grapevines and you want to know what's going on. Well, one of the ways that you probably use all the time in diagnosing a problem is symptoms. And symptoms are a great way to diagnose a problem. If you have a lot of experience and you know what's going on with your specific plants, you might be able to see something and know right away, hey, this is leaf roll. And you can make management decisions like that, or you may send your samples off to a lab to confirm, or you may not know what's going on. Another technique that's really commonly used is ELISA. And the way that ELISA works, ELISA is a great technique for analyzing thousands of samples. It's really fast and really inexpensive to analyze tens of thousands of samples at a time. The way that it works is a scientist somewhere purifies a virus or another material. They inject it into a rabbit, and the rabbit makes antibodies against it. Then you extract those antibodies from the rabbit. And when your sample comes in, what happens is it gets spread out on a plate. And that purple thing there would be the virus, the very specific virus that was injected into the rabbit. Then the green Y-shaped thing would be the antibody that the rabbit makes. And if you have the exact thing that the rabbit was trained on, then that antibody will stick. Then there's an anti-rabbit antibody that's in blue that has a chromophore on it. That gets washed over, that sticks to the rabbit antibody. And then an enzyme or something that activates the chromophore gets washed through, and you get a signal. So the important things here are it works. If you have the exact thing that it was trained on in high enough quantity, you're going to get a positive. There's no amplification here. So you have to have enough of the virus or whatever it is of interest to get a signal. But it's a great, quick and dirty, inexpensive way to test for something like red blotch. Next is PCR and qPCR. PCR stands for polymerase chain reaction. qPCR is quantitative polymerase chain reaction. And they're essentially the same thing. The difference is that qPCR is read by a computer, so it makes it more sensitive. They start with your plant material. It gets extracted, and nucleic acids, RNA and DNA, are extracted from it. And then each of these pieces of genetic material has a particular shape, has a different. There's 4 letters, right, that make up our nucleic acids: A's, C's, T's, and G's, and they're in a very specific order. And if somewhere someone has purified a virus or they know what that virus is, they design these primers that have the specific order in mind. And when you have the exact match, it sticks. So in the bottom here, you can see the polymerase chain reaction. You start with, if you start with DNA, you start with two double-stranded pieces. That's the green at the bottom. Those are heated, and then they separate into two pieces. The red is the primer. If it's an exact match, the primer will bind. The reaction proceeds, and those pieces of DNA are copied. Those single-stranded pieces become double-stranded pieces. The process starts over. It gets divided, primers again. So you get lots of amplification, and you very quickly get a very strong signal for something that may have been pretty minor at the beginning, which means that if you have the exact match, you're most likely, you're going to get a positive, and you're going to get a report that says you have red blotch. So that's great. The problem is when qPCR and PCR may not work, which is that viruses can mutate all the time. And when you have a mutation, then the primers don't fit exactly, they don't stick, and you don't get amplification, and you don't get a signal, and you get a report that says you do not have red blotch, even though maybe you do. So here we have a bunch of sequences on the screen. Each of these sequences is a virus from a different plant, the same virus from a different plant. On the bottom, you have the one that the PCR primers were designed for in yellow. And if you look at the ones just above that, you'll see the next four going from the bottom up are exactly the same. Those primers are going to stick, you're going to get amplification, and you're going to get a positive. The one above that has a mutation where an A became a C, and that C is in red. Those are those blue vertical bars. In that case, it's not an exact match. You may or may not get amplification. Higher up, the next one, you have a mutation, an A became a G, and you have this mutation with the C where an A is a C. In those cases, you're probably not going to get amplification. You're going to get a negative, even though this virus is there. There's a couple like that, and then there's four where C became a T. Similarly, one mutation change, it depends on a few different things, whether or not those primers will stick, and you'll get amplification. So that's why PCR and qPCR may not work. It's a great tool if you're looking for something very specific. It's a great tool, and it can work really well. If you don't know what you're looking for, obviously, then you can't test for it with a PCR because you're looking for something specific. And if what you have is slightly different, if it has mutated, you're going to get a negative. So the next technique I'm going to talk to you about is next generation sequencing based. It's a metagenomics approach, and this is what we use at Root Applied Sciences. But I do want to point out there are next generation sequencing based methods that are not metagenomics based. They're amplicon based. So they use primers just like PCR and qPCR. Instead of just reading a positive or a negative, they sequence in between those primers, but they're still based on primers. What we're doing is metagenomics based. So we take your plant material, we extract the DNA and RNA just like for PCR. And then instead of using primers to stick and read something in between, we actually read all of the sequences. We send them to a sequencer, they get read, it goes through our proprietary bioinformatics pipeline, and you get a report that lists everything that you have in your sample. So we haven't had a sample yet that doesn't have a virus in it. As you guys know, there's viruses that you care about more than others. Some are probably not important. We also provide blurbs for each virus about what we know about it scientifically. So in our method, all nucleic acid is sequenced. There's no predetermined target, so there's no bias, but it's more expensive. Here are some of the viruses that we detect that have been found in grapevines before. All of these have been found in grapevines before. That doesn't mean that they're all going to be a serious problem, but there are a lot of them. So I just wanted to do an overview again of the difference between metagenomics-based next-generation sequencing and quantitative PCR. Both methods are extracting your nucleic acids. qPCR is amplifying a target. If your sequence does not match the target, or if there's not enough of it, it's not detected. Next generation sequencing using metagenomics reads the sequences directly. There are many fragments read for every virus. So PCR is using a primer that's targeting a specific part of the virus. It's trying to target something that's relatively conserved so that if there are variations, you're going to get them all. It's not perfect. But because we're reading all aspects and can identify the entirety of the virus and not just one piece, where our metagenomics-based next-generation sequencing is more sensitive as well as more robust. When is this testing helpful? If you think that you have fan leaf and you're not sure and you want to send it for testing, you should send it to whoever you send it to right now for testing. And if you get an answer and if it says it's not fan leaf and you really think it is and you want to send it to us, we'd be happy to test it for you. But we think there's probably other times when our testing is more useful. Pre-planting, it's great for making sure that you know what you're planting, what's in your planting material pre-planting. It's great for either mystery symptoms or when your test results are wrong. Something's going on, you're not sure what it is, it's weird, it's a problem, send it to us, we'll let you know what's going on. Sometimes it's a combination of viruses. Some of our clients are really interested in, we have one client, for instance, he has a lot of different blocks that they want to replant. So they wanted to know essentially the health status of each one so that they can plan which blocks they're going to replant when. For premium vineyards, some clients just want to know what's going on. Before you see symptoms, your plant has a virus, right? It gets the virus, it's asymptomatic, and then symptoms develop. Well, we can detect the problems when it's asymptomatic. We can detect the virus. The pathogen is already there. So if you have a premium vineyard and you want to know what's going on, you want to stay ahead of things, you can randomly send samples in and we can let you know what's coming through. So that's also like proactive surveillance for novel pathogens. Anyway, thank you for your attention. Any other questions?
So if you know you do have a virus before the symptoms appear, what do you do? Because you can't, So if you do notice you have a virus before the symptoms appear, then what's your next step?
Well, if you know what the virus is, that can inform your management, right? So if you know what the virus is, we also give you information on vectors. So you could yank that. You might want to test the surrounding vines. The other part of our company is working on developing, essentially, if you have a problem here, where else do you likely have that problem? So you can yank that vine. You can look at the vines nearby. You can control the pest that may be transmitting the virus. There's a number of things that you can do. Ultimately, it's your choice, but certainly you can try to get ahead of it.
I was wondering what a single test would cost.
It's between $1,000 and $2,500 per test, and that's based on the number of samples you're submitting and the turnaround time.
Mm.
That's for NGS metagenomics.
And what about for PCR?
We don't do PCR.
How many samples would you need per acre, or to diagnose a vineyard? Because you're only sampling each vine, right?
Typically we'll take a couple petioles from the same plant. We can also do cane, of course, for pre-planting. But we're going to tell you what's in your sample. So if you gave us one petiole, we'll give you information on that petiole. If it has a virus in it, you'll get a list of all the viruses in it. I haven't found one yet that doesn't have a virus. I'm sorry? We can do a composite sample. We don't, so compositing 2 samples is easy. We can composite up to 4. We can talk to you more about the specifics of what you want. One of the things I didn't mention here is another use. We're talking to someone, he wants to send us samples from an entire subset of a nursery block. So hundreds of samples. But we're talking about how to composite them, how often to sample them to get the most information that's reasonable cost. The problem is when you composite a sample, when anybody composites your sample, when we composite a sample, we actually do an extraction on each sample because you never have the same extraction success on a different sample. And so our product is a premium product. We want to make sure that we're giving you information on every piece of it. So we don't composite the sample until after the extraction so that we know that we're going to get an equal amount of coverage on each plant. So that costs us money, and so we can't do it for too much. Yeah.
So my question is on endophytes, like bacteria maybe that are in the vine. How do you separate those out from the virus DNA that you're looking for? Is it pretty easy? The differences in DNA is pretty significant, so you can kind of cast those aside?
We read the bacterial DNA sequences too, and we can give you a list.
Have you looked at all about strain identification? I hear there's some non-pathogenic strains of Pierce's disease, and there are some pathogenic strains. Are you able to tell those apart?
The short answer is yes, we can identify everything at a very fine level, including even substrain. The longer answer is you have to talk to Rodrigo. If it's Pierce's disease specific, obviously I'll have you talk to Rodrigo Almeida because he's our co-founder and he knows all about Xylella fastidiosa and Pierce's disease. So. All right.
Thank you very much, Sarah.